Okay, now today we're going to be discussing Chapter 18, or the immune system, and this is actually the last chapter of the course, so congratulations, you made it. So here it goes. Okay, so the topics that we're going to be talking about, we want to talk first about the innate or the general immune responses, and then we'll talk about the more specific, the adaptive immune responses. 18.1, like the immune cells and cytokines, those are going to come as we talk about these different techniques. And then we are going to be discussing things like to get an idea about HIV and AIDS, and then the harmful immune responses. So we'll start off with the innate immune responses, and those are the responses that our bodies can provide to anything that is trying to invade it. Okay, they're very general, non-specific things that our body does in order to prevent things from coming into the body, or if it does, then, like things that kick in order to fight it off, okay? And these, again, are very general, meaning that it doesn't care if the invader is a virus or a bacteria or a yeast infection or a parasite. These are going to kick in regardless. So again, they're innate, they're non-specific, they're very general, and those are made out of your defenses at the body surfaces. Okay, so your body surfaces, intact skin and mucous membrane, are regarded as a mechanical barrier and your first line of defense. Then, if things are able to invade that barrier, then other things, then the body has to defend itself. So we've got an inflammatory process, interferons, which are different chemicals that the immune cells use to communicate together. There's fever, there's phagocytosis, and then there's the complement system. So we want to talk about these different things. So first off, the defenses at the body surface, and again, that's your very first line of defense. So the best thing the body could do is to prevent pathogens from entering, and that is by these mechanical barriers, your intact skin and mucous membrane. So you can see here, so what happens if the skin is not intact? Okay, so you can see here in this image, there's a splinter, and now the skin has is interrupted. And that splinter, not only did it interrupt the skin, but it actually introduced bacteria into the body. So what happens is that your first responders are going to come in. You see here, this is where your blood cells are, and the first responders of your blood system are the neutrophils. So neutrophils are the first cells that are going to arrive to the area. They are phagocytic cells, which means that they are going to engulf and eat up the bacteria in order to fight it. You see these white blood cells, or the neutrophils, exiting out of the blood vessels through a process known as diapedesis or diapedesis, which is basically these cells kind of squeezing through the capillaries. Now, what made these capillaries wider? That is the whole inflammatory process. So you see the capillary here is normal sized capillary. Once an area is damaged and chemicals are released by those damaged cells, that is going to lead to the dilation of this capillary. So you see here that this capillary is now wider, the pores are bigger, and that is going to allow these white blood cells to leave, kind of squeeze themselves out of these pores. Neutrophils, again, are your first responders. They remove the cells, or they remove that bacteria in any foreign body by phagocytosis. And then fibroblasts are going to come in from the skin, and those are going to start off the repairing process, leading to fibrosis or a scar tissue. Okay, so that is kind of like, in short, what happens if there is an interruption in the skin or mucous membrane. Now, these are different chemical mediators. Okay, so, there's a whole list of them, and I'm going to kind of focus on some of the more important ones. So we have the complement system. Okay, and the complement system is a whole bunch of proteins that are found in the body, in our blood, and they're made out of plasma proteins. So these plasma proteins are made in the liver, and they are circulating in the bloodstream, and they are not activated unless there is a need to. So they are inactive. Complement is kind of like the so let's say you have an invader trying to get into your house, okay? You try to stop it off, and if you can't, you're going to call the police to do it for you. So you call a professional to do that. Okay, so complement is considered like that police officer coming in, okay? They're more professional when it comes to fighting off pathogens. Their selected functions are opsonization, or they directly kill the pathogen. So opsonization means that if they can't kill the pathogen themselves, they red flag it and make it easier for the immune system to identify them and attack them. Okay, there's going to be more about the complement system as we go. Histamine that is made by mast cells and injured cells are going to produce histamine. Histamine is a vasodilator. So we've seen in the previous slide how the blood vessels dilate in order to bring in more blood and help with the white blood cells to reach the damaged area. We also have cytokines. Cytokines, and you notice that they're plural because we have lots of those, and those are made by activated immune cells. And these cytokines attract more leukocytes. Okay, so they perform a process known as chemoattraction for leukocytes. So you'll find that some immune cells or some white blood cells, when they are activated, they are going to make cytokines, and cytokines are going to attract more leukocytes. And they kind of work like these as paracrine hormones. Okay, so these are kind of the ones that we want to focus on. You can go ahead and read and kind of run through the other mediators. So this is the process of phagocytosis. You see here one of the phagocytic cells, and the phagocytic cells are mainly macrophages, neutrophils, and monocytes. Okay, so a couple of things to keep in mind. Macrophages are the same pretty much as monocytes, okay? If that cell, when it's inside of the blood vessel, we call it a macrophage. When that macrophage leaves a blood vessel and is now in the tissue, it is now called a monocyte. Okay, both of them are highly phagocytic cells. Neutrophils, as we said, those are your first responders, and they are highly phagocytic as well. So your phagocytic cells are the M&Ms, monocytes, neutrophils, and macrophages. So let's talk about the process. You can see here, this is one of the phagocytic cells. That is the microbe that is going to be engulfed through endocytosis. And you'll make a phagosome, which is the soma as a body, and the phago means, what has been eaten. And inside of those cells, there are lysosomes, and these lysosomes are full of digestive enzymes and hydrogen peroxide, and they are going to literally kill and digest that bacteria or that microbe. And you can see it here now. That the microbe has been killed and digested into small particles, and then those particles are going to be released either into the cell or out of the cell. Okay, so that is how the process works. So the lysosomes, again, these are full of lysosomal enzymes and hydrogen peroxide, and those help kill and digest the microbes. Now, what is the function of these phagocytic cells in the innate immune response? So when the phagocyte is intact or comes in contact with these microbes, it is going to eat it up by phagocytosis, as we've seen in that previous slide, and then it leads to the intracellular killing of the microbes. But it is also going to lead to release of different chemicals. Okay, so now that this one cell figured out or identified a foreign body, it has to let the other immune cells know about it, okay? So it's going to release some chemicals in order to do so. So it is going to release chemicals that increase the inflammatory process. So you are going to release like chemicals that are going to lead to vasodilation, for instance. It is also going to lead to the extracellular killing of microbes. So it is going to activate some of the cytotoxic T cells or the antibodies, maybe, depending on what kind of microbe we're talking about. So it is going to activate other cells to help in the extracellular killing of the microbes. So the intracellular killing was that phagocytosis, okay? So there are other ways of killing microbes outside of the cell that we're going to be talking about later on, and we call those extracellular killing. It is also going to activate the clotting and anti-clotting pathways. So you want parts, in order to keep the infection in one part of the body, you might want to form, or your body might want to form some blood clots, so to kind of make a barrier between the site of the infection and the other parts of the body. But at the same time, you want anti-clotting pathways because you want to have it release blood thinner like heparin in order to bring more blood into the area in order to help with the inflammatory process. And then we have the hormonal regulation of the body response to the infection. So you have other things going on in the body. So you have, fatigue, muscle fatigue, maybe muscle weakness or muscle ache, muscle pain. There might be loss of appetite. There's fever. So all of those are regulated by different chemicals released by these phagocytic cells. Now to the complement system. Like I said, the complement system are more organized. They are a whole family of plasma proteins. They are made by the liver and they are inactive. Okay, in order to activate them, you activate one, and then that, let's say you activated C1. Okay, so there's about 30 of them. When you activate C1, C1 is going to activate C2, and so on until you reach the end product. And your end product is going to make this huge complex of a protein known as the membrane attack complex. And the membrane attack complex does exactly what the name says, where it is going to attach to the plasma membrane of the bacteria or the virus, and it'll start making little holes in the membrane. Okay, so it'll attack the membrane or the protein coat of the virus, making holes in it, and that is going to obviously destroy the microbe. There are two different ways to activate complement. Okay, so you can either start activation from C1, okay, and that is known as the classical pathway. So know that C stands for complement, and because we have about 30 of them, we number them. So C1, 2, 3, all the way up to 30. If you activate from C1, that is known as the classical pathway. In order to do so, you have to have an antibody to start this pathway. But we have alternative ways to do that, to activate complement, and these are known as the alternative pathway, where you do not need an antibody, and that starts the process at complement 3. So you skip 1 and 2, and you go ahead and start at C3. C3 is the most important protein in this whole cascade. If you activate C3, that automatically means that you are going to eventually make a membrane attack complex. C3 has a little portion, tiny part in it known as C3b. C3b is going to be added onto the surface of the antigen and acts as an opsonin. So whenever you hear that word opsonin, you kind of want to think of a red flag. Okay, so C3b is that red flag that is going to attach to the surface membrane of the antigen, and that is how phagocytes are going to recognize that this is a foreign body. So we'll take a look at the different functions of the complement system. It'll directly invade the microbes by the membrane attack complex, and as we said, it'll make tiny holes in the bacterial wall or in the virus coat, and that is going to leak, definitely leak all of the stuff from the cell and definitely end up in destroying and killing that cell. Complement also helps in the vasodilation process, and that increases the permeability of the capillaries. It is a chemotactic substance. Again, chemotaxis means to kind of bring in or call in more cells to help. And then it enhances phagocytosis through opsonization. So again, opsonization means that we are now red flagging this pathogen, this foreign body, in order to make it easier for the phagocytic cells to identify. And this, you see here how that works. So this is your bacteria. And on the bacteria, the complement system left this little flag, that red flag that we called the C3b. Again, C3b is a little portion from the complement, from the third complement in that cascade. And it leaves that red flag on the bacteria. Now your phagocytic cells are able to identify C3b because they have a C3b receptor. So the C3b is attached to the bacteria, but the C3b receptor is found on the cell membrane of the phagocytic cells. Now when those attach, again, it helps in the process of phagocytosis. It makes it easier, say if this was a macrophage, it makes it easier for that macrophage to identify the bacteria as a foreign body because it found the C3b on its surface. Now we've got different chemicals that come out. One of those chemicals are, or some of these chemicals are known as interferons. So interferons are with an S again because we have several types of them. And we're going to be really talking about two of them in this chapter. So the first one to talk about is the type 1 interferon. You'll find it maybe sometimes in the slides as IF. IF is just short for interferon. So what is the function of interferon? First off, it helps generate a fever. It is considered an endogenous pyrogen. So pyrogen means it generates fire, and endogenously, meaning that inside of the body. So it is going to change the thermostat in the hypothalamus and make your body temperature go higher. That helps in two different ways. First off, it makes that pathogens usually need your normal body temperature in order to thrive. So when you make the environment too warm for them, that is a way of killing them. And the second thing, that when your body has a fever, it hides all of its iron. Okay, and a lot of the bacteria and a lot of microbes need iron in order to thrive. So hiding the iron and elevating the temperature is a way for your body to kill these pathogens. Okay, it also interferes with the abilities of the viruses to replicate. So type 1 interferon is considered an antiviral, and it also kills tumor cells. So you really want to keep that in mind, because one of the, some of the research done nowadays, whether it's on how to treat different viruses or how to kill tumor cells using, our own immune system, a lot of this research involves the type 1 interferon, or interferon 1. So let's take a look at how it interferes with the ability of the virus to replicate. Okay, so know that a virus is an obligatory intracellular pathogen, meaning that in order to survive, it has to live inside of a cell. So you see here, this virus invaded the cell. Now this is an infected cell. Now this infected cell knows that it's dead, okay, and there's no hope for it. What it does, it releases the type 1 interferons in order to let its neighbor cells that are still healthy into to kind of warn them and protect themselves so that this virus has no way of invading them. So now these healthy cells make an antiviral protein, okay, and now that this virus tries to go in, into this neighbor healthy cell, that antiviral is going to prevent the virus from replicating. So even if it's still able to invade it, it prevents it from replicating itself, prevents it from making copies of itself, and so on. So again, that's why a lot of the research done nowadays, whether it's on how to treat different viruses or how to, treat different tumors, cancerous tumors, is really directed towards interferon 1. Now we're going to talk about the adaptive immune responses. So what we talked about previously, these are very general, non-specific things that your body does in order to protect itself. Your third line of defense is an adaptive process, meaning that some things, these non-specific things are not enough. Okay, you have to provide more in order to protect yourself. And that is known as the adaptive immune response. Or this is what we consider, if you activate this response, now you are immune to these diseases. So we have the primary lymphoid organs, and that's the bone marrow and thymus. Okay, know that the bone marrow, that is where all of the hematopoietic cells are, and we make, if you guys remember the that slide from the blood system, I'm not sure what chapter that was, but we talked about, the T lymphocytes and B lymphocytes, and we said that the precursors for B lymphocytes are made in the bone marrow, and they mature in the bone marrow, while the precursors for T lymphocytes are made in the bone marrow, but they have to go into the thymus and they mature there, hence the name T lymphocytes. The maturation process is helped with the hormone thymosin that is produced by the thymus gland. The thymus gland is a gland that is very active in young kids, and that is how their immune system is developed. As we age, the thymus gland gradually shrinks and disappears, and until it's totally replaced with fibrous tissue and scar tissue. And that is why in seniors, there is that high incidence or they are at risk of infections, of getting infections because of the degradation or the disappearance of the thymus gland. Okay, so those are your primary lymphoid organs. We have secondary lymphoid organs, and those are the spleen, lymph nodes, the tonsils and adenoids. And then we have like lymphocytes that are kind of scattered throughout the body. If they are gathered together in relationship to areas that are lined with a mucous membrane, we call them mucosal associated lymphoid tissue, or MALT. So we have these little aggregations of lymphocytes that are found in the intestine, respiratory tract, genital, and urinary tracts. And you'll see that all of these, open up to the outside world through different parts of the body lined by mucous membranes. Okay, you'll see that the T and B lymphocytes might be made and mature in these organs, and then they migrate into the rest of the body. So they'll migrate, and some of them will remain in the spleen, others in the lymph nodes, tonsils, and in the malts. So let's talk about the derivation of the B cells and the T cells. Okay, so it was right here. So we have in the bone marrow, remember we talked about the hematopoietic cells, and we have the lymphoid stem cells and myeloid stem cells. The lymphoid stem cells are the ones that are going to eventually give you your T and B lymphocytes. The myeloid stem cells give you all of the rest of the cells of blood. So your lymphoid stem cells, if they mature in your bone marrow, they are going to give you mature B cells. Okay, if the cells, though, differentiate and then migrate to the thymus, where they mature in the thymus with the help of thymosin hormone, this is where we make the mature cells. T lymphocytes, we have different kinds of T lymphocytes. The ones that we kind of are going to be focusing on in this chapter are your helper T cells and your cytotoxic T cells. Okay, as you see here, all of these are mature cells, okay, but they are not active. Okay, so they are mature, they are ready for activation, but they have not been activated yet. These cells are going to be activated only in the presence of an antigen. Okay, so they are ready to work, but they're not going to do anything unless there's an invasion by an antigen. So you see here that these mature B cells and the mature T cells through the bloodstream are going to lodge themselves in the secondary lymphoid organs, which again were your spleen, lymph nodes, the MALT tissue, and the adenoids. And then again, when they are activated by an antigen, the B cells change into plasma cells, and plasma cells are antibody factories. Okay, and these antibodies are going to attack the antigen. Okay, B cells do not do the attacking themselves. They change into these plasma cells, and plasma cells secrete or release antibodies, and the antibodies are the ones that attack the antigen. This kind of immunity is known as humoral immunity, and that is the function of B lymphocytes. While the T cells, on the other hand, these cells are kind of more aggressive cells and they do the killing on their own, okay? So they do not make antibodies to do the killing for them, but the T cells are more aggressive. They attack the antigens, and that is known as cytotoxic immunity. And the cytotoxic immunity is the function of T lymphocytes. Now here's a summary of how all of these cells work together. Okay, so let's kind of take it from the very top where we encounter, or our immune cells are going to encounter an antigen. We have B cells, helper T cells, and cytotoxic T cells. Remember again, these are all mature, but none of them are active. What has to happen, or what is going to happen, is that the B cell, cytotoxic T cells, and the helper T cells, they have the ability to identify that there is an antigen. Okay, so they can encounter and recognize the presence of an antigen. But your B cells and cytotoxic T cells cannot activate themselves. Okay, so cytotoxic T cells and B cells have the ability to figure out that there's an antigen, but they can't do anything unless they are activated by the helper T cell. Now, your helper T cell releases cytokines, which again are paracrines, chemicals that are going to activate this cytotoxic and this B cell. Now the B cell is activated. It'll change or start making these plasma cells, and plasma cells again are your antibody factories, and antibodies are going to attack the antigen. If we're talking about this cytotoxic T cell, that again is activated by cytokines from the helper T cell. And these cytotoxic cells, even from the name, okay, it tells you that I am toxic to cells, okay? So they can directly attack the antigens. So the most important cell in this process is really the helper T cell. If the helper T cell is not activated, it will not be able to activate your B or your cytotoxic T cells. And the helper T cell is the aim of the HIV virus. It will attack the helper T cell. So the helper T cell is kind of like the person that helps in the back, while the B and the cytotoxic T cells do all the work. Okay, but really the cell that makes it all happen is this helper T cell. So if you really want to damage the whole immune system, this is the cell that you need to damage. Now we'll take a look at a lymph node. We have their lymph nodes are scattered all over the body, and you have the lymph that goes in through one end. So that would be your afferent lymphatic vessels. Lymph goes in, and you have these different pulps. You have white pulp and the red pulp. Those are full of either T and B lymphocytes, and these are going to act as filters. And the lymph is going to exit through the efferent lymphatic vessels, and then eventually will drain into the venous circulation and then back into the systemic circulation. So how do these adaptive immune responses work? Okay, first off, you have to have an antigen, okay, and that antigen has to be encountered and recognized by your lymphocytes. And we said that whether it's a B lymphocyte or a helper and cytotoxic T cells, they all have the ability to encounter and recognize the antigen. But then you have to activate the lymphocytes. Okay, so we said that your helper T cell is the cell that is going to activate the B and the cytotoxic T cells. And then we can attack the antigen. So depending on which cell, the B lymphocytes change into plasma cells, and your cytotoxic T cells are going to directly attack the antigens. So in order to identify and recognize an antigen, we need to understand that our cells not the pathogens, okay? Our body cells have, on our cell membranes, we have a complex known as the MHC, or the major histocompatibility complex. This is a complex that, or a protein that is coded by a gene found on chromosome 6. And all of our cells have these complexes, okay? And this is kind of the fingerprint of your cells. None of us have the same MHC unless you have an identical twin. So you would have, that you'd have identical MHC proteins as well. But even non-identical twins do not have the same MHC. So the only two individuals that would have that exact same complex would be identical twins. And MHC proteins are really considered your identity tags, okay, which is basically your genetic marker of biological self. This is how your immune system knows that this is your cell and not a foreign cell. We have two different kinds of MHCs, okay? There's class 1 and class 2. Class 1 MHC proteins, these are found on the surface of every single cell in your body with the exception of the RBCs, okay? RBCs do not have that class 1 MHC. They have, antigen A and antigen B and so on. So again, your class 1 MHC is found in all of your cells except for your red blood cells. While class 2 MHC, these are the cells that are found on just a number of cells. So for example, on macrophages, on B lymphocytes, and on dendritic cells. Dendritic cells are macrophages that work in the CNS. Okay, so your T cells, your T lymphocytes, have the ability to identify these different MHCs, okay? And that is known, this is part of the antigen presentation process. So when a foreign body comes into, or a pathogen is trying to invade the body, your immune cells are going to look at the different parts of it and try to figure out, is that the class 1 MHC of my body, or is this a foreign antigen trying to come in? So we'll look at this process further on in the next couple of slides. Now that we know the different kinds of MHC proteins, we want to figure out which MHC proteins interact with which cells. So the MHC, the class 1 MHC, which is the class that was found on all of our cells, okay, they're found on all of our nucleated cells except for your erythrocytes, those can interact with your cytotoxic cells, cytotoxic T cells. T lymphocytes, there's so many of them, okay, and they are named according to these different clusters, and hence the name CD. So CD stands for cluster of differentiation, and there are a lot of those. So the cytotoxic T cells are of these cluster of differentiation type 8, or they are known as CD8. So your cytotoxic T cells can only bind to the class 1 MHC, which means that any cell in the body that has, again, a class 1 MHC and figures out that there's something weird going on within its cell, usually it's a virus or a cancer developing, has the ability to show that antigen, or to show that foreign body on its class 1 MHC on the surface, and your cytotoxic T cell can identify it. Okay, your helper T cells, those are of the cluster differentiation 4, and those can only bind to class 2 MHCs. And remember that your class 2 MHCs are found on very specific cells, your B lymphocytes, your macrophages, and dendritic cells. So these are cells have to figure out a way to break down the pathogen and attach a part of it to its class 2 MHC, and that is how the helper T cells can find them. B lymphocytes do not interact with MHC proteins, and NK, or natural killer cells, these are killer cells these are very, very aggressive kind of cells These do not really interact with MHC proteins at all in order for activations. There are different ways to activate an NK cell. Okay, so let's take a look at the sequence of events in which an antigen is processed, okay, and presented by a macrophage. So let's remember that these macrophages have on its surface have the class 2 MHC proteins. When there's an antigen trying to invade the body or is now inside of the body, the macrophage identifies it as a foreign body. It will engulf it, and as it's engulfing it through phagocytosis, it engulfs this class 2 MHC that's found on the cell membrane. So normally this would be out here in the surface. But now that it has identified an antigen, it'll engulf the class 2 MHC protein with the antigen and break down the antigen into smaller fragments. Then it will attach one of those fragments to an MHC protein. Now that MHC is going to, it will be exposed on the surface of the macrophage, attached to the portion of the antigen. Now this helper T cell, so remember your helper T cells were of the CD4 kind, and they can only identify with class 2 MHCs. So normally when this helper T cell, is interacting with a class 2 MHC, but the class 2 MHC is empty, that means there are no foreign bodies. But in this case, there is an antigen attached to it, or an antigenic fragment attached to it. So now this helper T cell knows that there is a foreign body. Okay, and that is known as, the macrophage presenting the antigen to this helper T cell. That is why this macrophage is known as an antigen presenting cell, or an APC. APC again stands for antigen presenting cell. So again, the whole thought, the whole process really revolves about having an MHC complex, a class 2 MHC on the surface that is usually empty, but when an antigen invades, the macrophage will break it down into smaller fragments, and now this class 2 MHC has the part of the antigen attached to it. So when this helper T cell comes, it sees that the class 2 MHC is no longer empty. It now has a foreign body attached to it. And that is how this helper T cell is now activated. Okay, so this is the interaction between a macrophage and a helper T cell. Now, what about your B cells? Okay, remember we said that B cells do not even need, they don't interact with your class 1 or class 2. What happens is that we've got antibodies on the surface. Okay, so the antibody will attach to this specific antigen. It engulfs it and it breaks it up. And when it does that, again, it'll take a fragment of the antigen and attach it to the class 2 MHC. Class 2 MHC is now on the surface, and this, again, helper T cell will identify it as having a foreign body. Okay, so again, your antigen presenting cells, we talked about the macrophage and how it works. We talked about the B lymphocyte and how it works as well. Okay, it starts by the antibody recognizing the antigen. And then we'll talk about, what are the events that are required for the activation of the helper T cell. So now that the helper T cell figured out that on the class 2 MHC, there is an antigenic fragment. Okay, this whole cell represents your antigen presenting cell, which again could be your macrophage or a B lymphocyte or a dendritic cell. Well, we need the interaction between the class 2 MHC with the antigen attached to it with the CD4. But there is something else that we need, and that is known as a non-antigenic matching protein. These two proteins acts kind of like a co-stimulus. Okay, so it increases the activation, And this antigen-presenting cell releases interleukin-1 and another chemical known as tumor necrosis factor. Both of these are going to help in the co-stimulation of helper T cells. Remember, once the helper T cell identified an antigen and becomes active, now this helper T cell can release chemicals to activate B lymphocytes and cytotoxic T cells. Now, what about the cytotoxic T cell? Well, cytotoxic T cell can only interact, which is the CD8, has a CD8 on its surface, can only interact with class 1 MHC. Okay, the easiest way for me to remember which one needs which class MHC to interact is that when you multiply them, they have to equal to 8. Okay, I know it's a very cringy way, okay, but it just helps me figure out which one is which. So the cytotoxic T cell that has CD8 on its surface needs to interact with class 1 MHC. So if you multiply 8 by 1, you've got an 8. While in this case, your CD4 needs a class 2. So again, if you multiply the 2 and 4, you get an 8. Okay, so what about the cytotoxic T cell? Well, cytotoxic T cells, it does not need an antigen-presenting cell, okay? Remember, your antigen-presenting cells had class 2. But any cell in the body, all of our nucleated cells, remember, have class 1 MHC. So if any of our cells are invaded by a virus or have cancerous changes going on, these cells have the ability to present that antigen on its class 1 MHC. Okay, now these cytotoxic T cells have on their surface a CD8 protein. Those are going to identify the class 1 MHC and figure out, wait a minute, this class 1 MHC is abnormal. It has an antigenic particle attached to it. That means that this cell is either invaded by a virus or has cancerous changes and it needs to be destroyed. Okay, so you want to think of cytotoxic T cell as that cell that helps against viral, to attack cells that have viral infections or cancerous changes. And again, this is something else that is also being experimented in ways to kind of enhance or boost our own immune system to prevent viral infections or to treat viral infections and to treat cancers. So what are the summary events, in order to get a cytotoxic T cell to kill a cell that's has a viral infection? Basically what it does, it makes these little holes or perforations into the cells. Okay, so once that it identified this as an infected cell, the T lymphocyte, now this activated T lymphocyte is going to make perforins, which are chemicals that make literally perforations or holes into this infected cell. Now this infected cell is going to die, and that is how it helps in preventing the spread of the infection. Now for natural killer cells. These are kinds of lymphocytes that look very similar to cytotoxic T cells. Their major targets are also virus-infected cells and cancer cells. So you'll see that viral infections and cancerous changes to cells, these have like their own cells and own chemicals to help in their fight. So we talked about the cytotoxic T cells. You also have these natural killers. Natural killers do a survey of our cells all the time, okay? So what we have is that as we're sitting here, as we're healthy individuals, lots of our cells undergo cancerous changes, but our natural killer cells are always surveying all of our cells. And once they identify a cell that has cancerous changes going on, it will go ahead and directly attack and directly kill that cell. Okay, so, and it does not need class 1 or class 2 MHCs. It will recognize these specific antigens through different ways that are not dependent on class 1 or class 2. Now, what is the role of interleukin-2 and interferon-gamma in the process of activation of these different cells? So when we have an activated helper T cell, that activated helper T cell, we said that it's going to release cytokines. Cytokines is a very general term. A couple of these cytokines that are worth noting are interleukin-2 and interferon-gamma. These are going to further activate macrophages and natural killer cells. And when you activate these two cells, these cells are going to destroy target cells. Okay, so you will find that there's a lot of interaction between all of our immune cells. So remember, the macrophages were the cells to present the antigen to begin with to the helper T cell, but then your helper T cell goes in, releases interleukin-2 and interferon-gamma to further activate macrophages. So there's always this kind of like positive reinforcement going on between the cells where they are always further activating each other through different paracrines and different cytokines. Now, the development of immune tolerance. So how is it that our own immune system, our immune cells, are able to identify our own cells? Okay, so we have and that process is known as immune tolerance. How our immune cells, how do they tolerate our antigens? Okay, this develops during fetal life and in the early postnatal life to what is known as clonal deletion or clonal inactivation of cells. So clonal deletion, that's also known as central tolerance. That happens in the thymus and in the bone marrow, where basically your T and B lymphocytes are exposed to all of your antigens. Okay, so it's kind of like introducing your T and B lymphocytes to all of your antigens of your body. And that your T and B cells are capable now of identifying these self-antigens, and any cell that has the ability to attack your own antigen will undergo apoptosis, which is the process of self-destruction. Okay, so your own body, again, takes a look of all your T and B lymphocytes, looks at all of your antigens, and if any of these your own cells are going to attack, if any of your immune cells are going to attack your cells, it will undergo apoptosis. That is known as clonal deletion or central tolerance. But we also have peripheral tolerance, and that occurs in the peripheral organs or in the secondary lymphoid organs. That's known as clonal inactivation. That happens in your lymph nodes and spleen, where these self-reacting B and T lymphocytes, those are inactivated, but they're inactivated in the peripheral organs like the lymph nodes and spleen, and that's why it's known as peripheral tolerance or clonal inactivation. So let's take a look at the structure of an immunoglobulin. Immunoglobulins have 4 protein chains. So there's a light chain, 2 light chains, and 2 heavy chains. They are Y-shaped. Okay, the part of the Y on top is known as the specific antigen binding site. So these are variable between immunoglobulins, and they are specific to certain antigens, meaning that each antigen has its own antibody. While the constant region, which is this portion of the Y, that's a constant end, and it is not going to interact with the antigen. The part that interacts with the antigen is this variable end, where at the very tip it has these antigen-specific binding sites. We have 5 different kinds of antibodies. Okay, we've got IgG, IgM, IgE, IgD, and IgA. Okay, and for all these 5 classes of antibodies, you want to be able to answer these following questions. So we'll go over these, I believe the next slide. Let me check. Okay, so they are not on the slide, but you can go ahead and kind of write these in. So which ones activate complement? The complement system, remember, is that cascade that we talked about earlier. There are only two kinds, only IgG and IgM are able to activate complement. Now, which one of these are most abundant? IgG is definitely the most abundant kinds of antibodies we have, followed by IgM. Which one of these is found in exocrine secretions, for example, milk? That would be IgA. Okay, so IgA, or immunoglobulin A, is found in breast milk. Okay, and that is why it is important, especially during the first couple of days of breastfeeding, where it's rich in these IgAs, and that is transferred to the baby during lactation, and is really important since the immune system of the baby has not really kicked in fully, and it'll cover the baby for a couple of months up until the baby's own immune system is ready and able to perform its function fully. Okay, so found in exocrine secretions like milk, again, is IgA, but it's also found in other exocrine secretions. So sweat, for example, saliva, any exocrine secretion that you can think of would have an IgA. Now, which one of these attacks parasites and is involved in allergies? That would be IgE. And which one is involved in incompatible blood transfusions? So that would be the IgM type. Okay, and which one passes the placenta? That is IgG. Okay, so again, if anybody, during delivery, the presence of IgG in mom, okay, so certain antibodies that are of the IgG type in mom would pass to the baby and remain in the baby's body, providing some immunity up until, again, the baby's immune system is ready to perform its own function. So you have, the IgG that pass through the placenta, and then you have the IgA that is present in milk, and those, give the baby some kind of passive immunity until their own immunity is able to kick in and perform its own function. So what do antibodies do? Okay, so what are the function of antibodies? Well, they directly enhance phagocytosis, so they act as an opsonin. Again, these are red flags that attach to antigens through their antigen-specific sites, and the phagocytic cells are able to identify them easier. They also activate complement system, remember, which is only a function of IgG and IgM. They can also produce what is known as antibody-dependent cellular toxicity, meaning that you can kill some cells, but that are only in the presence of these specific antibodies. And they can directly naturalize bacterial toxins and viruses. So we have, not only do we make antibodies against antigens, but we're also able to make antibodies against different toxins, like, for example, tetanus. Okay, so the tetanus toxin, we can make tetanus toxoid, which is the antibody against the tetanus toxin. And we're also able to make antibodies against viruses. So it's not only, a bacterial thing, but viruses and even toxins as well. So let's take a look at how antibodies help in the opsonization process, or in the phagocytic process. Right here is the bacteria. Okay, and the bacterial antigen that is found on its surface is going to interact with the antigen-specific site of the antibody. Now, this antibody has its own receptor, okay, which is the receptor for Fc, which is the constant part. C starts for constant, the constant part of the antibody, and it will attach to that. Okay, and this is how your phagocytic cell, whether, again, it's a macrophage, neutrophil, or monocyte, is able to identify that this antibody is now attached to the bacteria, and it will go ahead and engulf it. Now, what about the activation of classical complement pathway by binding to the antigen? So remember, we said that the classical pathway, well, first off, we mentioned a couple things. So first off, that in order to activate complement, only our IgM and IgG are able to do that. So the GM type, and that We got two different kinds of complement. There's the activation. There is the classical one where you'll start off at C1, and that was dependent on the presence of an antibody, or you could activate starting at C3. Okay, so what we're talking about here is the classical activation pathway, where you need the presence of C1, that very first complement protein, in order to start the process. So when the antibody has identified the bacteria, okay, the antibodies have these sites known as a binding site for C1. It will bind to that very first complement protein, and that will start the activation process of the proteins as you go down the stream until you activate that C3b part, which again was your most important one to activate. And you'll also make that MAC complex, where that membrane attacking complex, where you basically are going to be making a pore or a hole into the membrane of the bacteria, and that will lead to leakage of everything from the bacteria and basically killing it. So that MAC complex can kill it, or that presence of the C3b that can attach to this C3b receptor, the phagocyte, and that could lead to a destruction in a different way. So for antibody production, there are two different ways where the body can make an antibody. First off, the body has to identify that there's an antigen. Okay, and then when it does identify the antigen and figures out which antigen is this, it can produce antibodies. That very first time it sees an antigen is known as a primary response. Okay, so when your immune system is exposed to the antigen for the very first time in its life, it needs time to process the antigen, identify it, and figure out which antibody do I need to make. That again process is a much slower one, and that is known as your primary immune response. But in the process, it'll make what are known as memory cells. These memory B lymphocytes, if you are exposed to that same antigen for a second time, now your immune system already knows it, okay? These memory B cells are going to recognize it right away and be able to produce antibodies right away. And that is known as your secondary immune response. So you can see here that when you're first injected with an antigen, so let's say you're taking a measles shot, okay? So you'll get a portion of the measles virus, and your body is seeing this for the very first time. It'll take some time to make antibodies. So you'll see here that it takes about, a little bit less than a month to reach the peak of the amount of antibodies that your body made to that first exposure to that antigen. And that again is known as your primary immune response. In the process of the primary immune response, you made memory B cells. So that let's say 10, 15, 30 years later, you are exposed to the measles virus for real now, and not in a shot. Your memory B cells that you made as a kid are going to identify the antigen, identify the measles antigen right away. And you see here that you are within, days able to make a secondary immune response. And you see that the secondary immune response, not only is it faster, but the amount of antibodies that are being produced are much higher than your primary immune response. Okay, so this is how, again, and all of the differences between whether the amount of antibodies being made or how long it takes to identify are all due to the presence of memory B lymphocytes here, but they are obviously not found in the primary immune response case. Now, I believe we're going to be talking about the difference of active and passive immunity. Active and passive immunity really means, where did the antibodies come from? If your body made the antibodies, that is known as active immunity. So your immune system was actively involved in making the antibodies. That is your active immunity. Again, meaning that your immune cells had to recognize an organism, had to activate different cells and produce antibodies. Okay, so now that it actively made the antibodies inside of your own body, that is your active immunity. That could you can get active immunity either from a vaccine, so just like in the measles vaccine that we talked about, or by an infection. So if somebody gets the measles as an infection, their body is also going to identify that measles virus and make antibodies against it, and they are also going to have active immunity. Vaccines, though, just because I mentioned this, is definitely a much better way because first off, you're not getting sick. Okay, you're not being exposed to the whole measles virus, so you're not actually getting measles. Plus, measles has very serious complications like meningitis or orchitis, which is inflammation of the testis in males that could lead to infertility. So it's a very serious disease, and it's not just about different spots that appear on the skin. Okay, there are lots of potential complications that could happen. So, but an active immunity can definitely happen either due to a vaccine or due to an infection. Okay, a vaccine, again, you'll get a small quantity of the pathogen, whether either it's a small antigenic part, whether it's a live pathogen or even the dead pathogen, or you can get a small amount of toxins. So remember, you're able to make antibodies against toxins like tetanus toxoid, for instance, and your body has to identify it, make antibodies, hence now you're actively immune against that particular organism. Now, if you are exposed to that organism again, you already have these memory cells and you can go ahead and protect yourself right away. So why is it that in some shots you have to take it every year, for example, flu shot, as opposed to measles? Well, the memory B cells of measles last for decades, while the memory B cells of the flu only last for a couple of months. Okay, so they pretty much kind of carry you through this winter, but then come next fall, you need another flu shot. Now for passive immunity, now that's a little bit different. Okay, passive immunity means that the antibody was made elsewhere, whether in a factory or in somebody else's body, and then it was passively transferred to you as pre-made antibodies. So those could be, for example, the IgG immunoglobulins that were made by mom and they were passed to the fetus through the placenta, or the IgAs that were made by mom and passed on to the fetus or to the baby during breastfeeding. Okay, so that is a kind of passive immunity that was given to the baby where these antibodies were made in mom and passively given to baby. Okay, again, it will cover this baby for some period of time, so it is an important source of protection for the first months of life until, again, their immune system is able to kick in and they're able to make their own antibodies. So what is the systemic response to infection? So we kind of talked about what does the immune system do and how it attacks different pathogens, but when we're sick or when we get an infection, our whole body is sick. So how do these different chemicals work in order to make us sick? So when we do have a microbe, okay, the monocytes and macrophages, those are going to secrete different cytokines like interleukin-1, tumor necrosis factor, and interleukin-6. Now that these are going to be found in our plasma, they perform different things, or they make us feel different things. So when they reach the brain, again, interleukin-1 is that endogenous pyrogen. It is going to change the thermostat in the hypothalamus. You'll develop a fever. It also changes your appetite. So most people have loss of appetite, decreased food intake, sleepiness, and fatigue. Okay. When these different chemicals reach the liver, the liver starts storing its iron. So remember, we said that a lot of organisms need iron to survive. So one of the ways to deprive organisms from iron would be by the liver keeping it. And it also keeps the zinc. And the liver is going to start secreting what are known as acute phase proteins. Acute phase proteins are used in the lab to, we can detect them by blood work to figure out, oh, you know what, this body actually has something going on. So for example, we'll find that there's a low iron and zinc levels, increased plasma acute phase proteins like elevated, what is known as a C-reactive protein, or an elevated ESR, which is an erythrocyte sedimentation rate, and other different labs. These chemicals will reach the bone marrow, and that will stimulate the bone marrow to make more leukocytes. So these sick individuals would have leukocytosis. The chemicals will go to the adipose tissue, leading to lipolysis, and hence there will be loss of weight and increase of the plasma-free fatty acids that could be used as energy, because notice that there is an increase in food intake. When these chemicals reach the muscle, they're going to start protein breakdown and increase of the release of amino acids, which can also be used as a source of energy. And then finally, these chemicals will stimulate the anterior pituitary to release ACTH, and ACTH will stimulate the adrenal cortex to make cortisol. And cortisol is going to be increased due to, remember, when you're sick, it is considered as a stressful situation to your body. So the hormone of stress is going to be elevated. So how can we kind of, things that we could do or avoid, some other things that alter your body's resistance to infections? Okay, so protein calorie malnutrition, which is something where individuals have malnutrition from protein. Okay, so their diet is really made out of starch and carbs and fat, while it is deprived of protein. Remember that your complement system, these are all plasma proteins. If you don't have enough amino acids, how are you going to be making them? So a lot of your immune responses, kind of depends on an adequate protein intake. The presence of a pre-existing condition, okay, that could also decrease the resistance to other infections. So let's say, for example, somebody maybe has diabetes or high blood pressure or asthma. All of these pre-existing conditions can really decrease the body's ability to resist infections. Stress, okay, can definitely decrease remember, stress increases cortisol levels, and we kind of talked about the effect of hypercortisolism when it comes to the immune response. Modest exercise, that is actually an immune booster. Okay, so it boosts your immune system and helps you resist different infections. Another factor that decreases immune function is sleep deprivation. Okay, so you really kind of want to, keep those in mind when somebody's kind of struggling with different infections. Now, immunodeficiency diseases, these result from, impairments in the immune system. So, for example, we have SCID, which stands for Severe Combined Immunodeficiency Disease. Okay, and this disease, in these individuals, there's an absence of both B and T lymphocytes, sometimes even natural killer cells, and this is a genetic disorder. The AIDS, or acquired immunodeficiency syndrome, that is caused by an infection. So unlike SCIDS, which is a genetic disorder, this is a viral infection. And remember, we said that the target of HIV would be the helper T cells. So basically, if you want to destroy your immune system, your target will be your helper T. Now, for harmful immune responses, we have graft rejection. So immune system is a good thing, but sometimes it goes overboard, and it can lead to some harmful responses like graft rejection, transfusion reactions, allergies, autoimmune diseases where now your immune system is attacking your own cells, or excessive inflammatory responses. So we're going to talk a little bit on tissue graft and organ transplantation, where your immune cells, again, are able to identify your own class 1 MHCs, but when you get a donor, if you get donor tissue, this donor has different class 1 MHCs, unless you're getting a transplant from an identical twin. Other than that, your class 1 MHCs are going to be different than your donor's class 1 MHCs. So the thought, the theory is that you want to make sure that you match these class 1 MHCs as much as possible. Okay, because before surgery, before transplant surgery, you want to give the recipient immunosuppressive medications. So to kind of, calm down their immune system and make sure that, or prevent the immune cells from identifying these, the donor's cells. Okay, and that will help in the decrease of tissue rejection. So some tools that we could do, we could, sometimes radiate or give drugs that will kill the actively dividing lymphocytes. Basically, what you're going to do is put this recipient into an immunosuppressive state. So one of the very effective drugs is known as cyclosporin. It doesn't kill the lymphocytes, but it blocks the production of interleukin-2 and other cytokines by the helper T cells. And remember, interleukin-2 was kind of responsible for activation of other cells. So when you do that, your helper T cells are fine. They do identify that there is an abnormal, that recipient cell is coming in, but it loses the ability to let the other cells know about it. We do have transfusion reactions, okay, which means that which could happen in an incompatible blood transfusion. So it's not really, we didn't really go into the details of blood groups, but I do want to kind of go over it really quick with you. So our blood cells, on the surface of our blood cells, we have different antigens, either antigen A or antigen B. And our plasma would have antibodies. If you are a blood type A, okay, your RBC would have antigen A and antibody B. You cannot have the same antigen and antibody because then they would be attacking each other, okay? So again, if you're blood group A, you'd have antigen A, but an anti-B antibody. Your blood group is named according to the antigen that you have. If you're blood group B, you would have antigen B and antibody A. If your blood type is AB, you would have both antigens on the red blood cells and no antibodies. If you're blood type O, you would have no antigens. That's where O comes from, 0 antigens. But you would have both antibodies, antibody A and antibody B. Okay, so in the process of an incompatible blood transfusion, it is due to antibodies and not the cytotoxic T cells. Cytotoxic T cells were kind of responsible for tissue rejection. Antibodies, and to be specific, it is the IgMs that are responsible for incompatible blood transfusions. And it really depends on the ABO system, the antigen A, B, or the absence of these different carbs that are found on the surface of your red blood cell. Okay, so you want to know the different antigens and antibodies that are present in each blood group. Okay, another antigen that's also important is the Rh antigen. Rh stands for rhesus factor. Okay, and it was first identified in monkeys. These monkeys are known as rhesus monkeys, and hence the antigen was named after them. And then we found out that we have the same antigen, but we kept the name. You might find it in some textbooks as antigen D, but they're the same thing. Most individuals do have the antigen, so would be Rh positive, and that's where the positive and negative comes in your blood group. So let's say, for example, if I'm blood group O positive, that would mean that I do not have antigen A, I do not have antigen B, that's why I'm a blood type O. Positive means that I do have the Rh antigen though, okay? The Rh factor is really important during pregnancy because it is a dominant factor, meaning that if mom is Rh negative and dad is Rh positive, the baby is going to be Rh positive. Okay, now this mom has never seen this Rh antigen before because she is Rh negative, but the baby is Rh positive. During delivery, there is a little bit of mixture of blood between mom and baby. Now that happens, the mom's immune system for the very first time in her life is seeing this Rh antigen, and it will react to it, making anti-Rh antibodies. That first baby, though, is going to be delivered, and the first baby is fine. But if the mom gets pregnant again, and the second baby is also Rh positive, well, now mom's blood has antibodies against that the second baby's red blood cells. Those antibodies are going to be attacking the second baby's red blood cells, leading to their destruction. And that is a disease known as erythroblastosis fetalis, or the hemolytic disease of the newborn. Now going over to the major types of hypersensitivity, there are 4 different kinds. There's an immediate hypersensitivity reaction, and those are mediated by the immunoglobulin E's, mast cells, and eosinophils, which is basically you touch something and you are allergic to it, and and you get a rash right away from it. Okay, so that is your immediate hypersensitivity. It is usually, though, more kind of like an antigen that you inhale and can lead to the IgEs, mast cells, and eosinophils are going to release different chemicals, and for some individuals, it gets hard for them to breathe and so on. That's the immediate hypersensitivity. It's something that happens right away, right after the exposure to the antigen. Sorry, the allergen, I should say in this case, because it's an allergy. Cytotoxic hypersensitivity, that is mediated by antibodies, and those are going to lead to the destruction of different cells, just like the hemolytic disease of the newborn that we just discussed, or transfusion reactions. You have, again, the antibody, the pre-made antibody. Now you have the antigen. They are going to combine, and the antibody will destroy that cell. In immune complex hypersensitivity, this is basically the whole process of autoimmune diseases, or some autoimmune diseases, but not all of them, okay? It is mediated by antigen-antibody complex that is deposited in tissue, and then that will lead to tissue destruction. So, for example, in RA, which stands for rheumatoid arthritis, or SLE, that stands for systemic lupus erythematosus, or just basically lupus for short, or rheumatic fever. All of these are different examples. So we'll talk about, let's say in the process of RA, for example, okay? RA and SLE, they're sometimes triggered by things totally unrelated. So you have they're more common in females. So you have this lady that comes in, she'll tell you that she had a viral infection, say about, a month ago, and then a month later, she started to develop pain in the joints and so on. Okay, what happened is that virus led to the production of an antibody, and then the antigen-antibody complex deposited in the joint tissue, leading to joint destruction. And that is what triggered rheumatoid arthritis in this patient. Okay, this is just one of the theories. There's so many other theories in RA and SLE. Okay, but this is the theory that that shows, the scenario of the immune complex hypersensitivity. So it is mediated by an antigen-antibody complex. Delayed hypersensitivity, that's mediated by the helper T cells and macrophages, and these do not depend on the presence of antibodies. So you'll see here that the first 3 kinds depend on antibodies. The only one that does not is the delayed hypersensitivity. So, for example, contact dermatitis by poison ivy. So on your first exposure, nothing happens. Okay, it's not until the second exposure that now these activated helper T cells and macrophages are going to lead to a hypersensitivity reaction, and hence the name delayed hypersensitivity, because it does not occur in the first exposure, it'll happen on the second one. So know that the difference, in this one is that it's delayed, and it happens by the T lymphocytes, while the other three are dependent on antibodies. So what are the sequence of events in the immediate hypersensitivity allergic reaction? So when you're first exposed to and that was the type 1 in the previous slide, when there is the allergen that comes in attaching to the antibodies that were of the IgE type, the IgE are found on the surface of a mast cell. Mast cells have all of these vesicles, and these vesicles are full of histamine and heparin. When we release all of that histamine in particular, so you want to think of somebody that has nut allergies, for example. Okay, as soon as if they eat it or smell it, or pass by somebody that is eating nuts, that allergen attaches to the IgEs that are found on the surface of these mast cells. The mast cells are going to be activated and release heparin. Now that heparin is going to lead to massive vasodilation, leading to the swelling and the edema, can even lead to difficulty in breathing, can lead to hypotension and shock, and can lead to massive production of mucus, leading to it making it very hard to breathe, and itchiness. Okay, so that is, how this occurs. You can see here, this is a mast cell, and you can see how rich it is in the secretory vesicles, having massive amounts of histamine. In anaphylaxis, that would happen, that is an allergic, one of these symptoms, it's a severe symptom of allergies, where you have massive amounts of these mast cells that release lots of histamine. And when that histamine enters into the circulation, that will lead to systemic vasodilation, okay, and bronchial constriction. Both of those together, the massive vasodilation leads to hypotension, and that is known as anaphylaxis. So the anaphylactic shock would be due to the circulatory and respiratory failure. Circulatory failure due to the massive dilation of the blood vessels, leading to severe hypotension, and respiratory failure will be due to the bronchiolar constriction, causing dyspnea, or if it's severe enough, it can lead to respiratory failure. An autoimmune disease is the inability of the immune system to identify your own antigens. Okay, so now remember we talked about, immune tolerance and how our immune cells are supposed to be able to identify our own cells. Well, something happened where now that is not the case anymore. So the immune attack that could be mediated either by the T cells or the auto antibodies, okay, can lead to these autoimmune diseases. So for example, these are all examples of autoimmune diseases. So type 1 diabetes, where there are auto antibodies against the pancreatic cells, rheumatoid arthritis and SLE, MS, which is multiple sclerosis, or myasthenia gravis. So in MS, there is autoantibodies against the connective tissue. In myasthenia gravis, there would be autoantibodies against the Schwann cells, or against the myelin sheath. And in this lady here, you can see the very characteristic known as the butterfly rash, or the malar rash, that happens on the cheeks and on the bridge of the nose, that is very characteristic to SLE, or systemic lupus erythematosus. So what are some possible causes of autoimmune attack? What is it that, your immune system was fine, and then all of a sudden it cannot identify your own cells? Well, there's so many different possibilities, which means that we're not really sure what is the exact cause. Or is it that everybody has a different reason? So it could be due to failure of the clonal deletion that happens, that we talked about, whether that should have happened in the thymus, or the clonal activation in the peripheries. Okay, is it that this individual was fine, but they got exposed to different drugs or in different environmental chemicals, and that led to the inability, the damage to parts of the immune system. So now they think of your self protein as foreign protein. Or was it that the person got a viral infection and that kind of triggered the whole process? Maybe there was some kind of genetic mutation that happened in the body. There's just so many different theories out there that, again, it's very hard to pinpoint what is the exact cause. These are the different cells that mediate the immune response. So, you can kind of take a look at it. There are this table, and then there's a table for the cytokines. I just put, these are copied and pasted from your book, just put them up there. So because we talk so much about these different chemicals and about the different cells, but if you want to kind of be able to focus and, know that, okay, so what does this cell in particular do? You can find them in these next floor slides, but we've talked about all of them throughout the whole course. And this finalizes chapter 18.